Hello!
Hi there.
Welcome to Microbiology Journal Club. We nerd out big about all things small.
My name is Danny, and in previous life I dropped out from my PhD in microbiology. Then I was a fact checker for pharmaceutical advertisers. Nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based at NYC, dedicated to aim at the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology, where I mostly work on bacteria. Since then, I've worked as a research integrity specialist, and I currently work for a scientific journal.
Every other week, we meet to talk about microbiology. On a typical day like today, we do an overview, well, today's not a typical day, but on typical days, we do an overview of the coolest microbiology papers that we've seen in the last 2 weeks. And this episode, we're doing a top 10 countdown of the top cited papers in 2022.
That's right. This week, we are doing a look at some of the most impactful papers that were published last year, notably the ones that don't involve COVID or monkeypox, because we've talked about that a lot. So this is an opportunity to talk about papers that we wouldn't ordinarily talk about. And we also want to do deep dives into viewer-selected papers. So leave us a comment if there's any paper you want us to do a much deeper dive into, and we'll look into making that content.
You can follow along with the papers that we discuss on either week in our shared Zotero library. Our social media handles and the link to the library are in the doobly-doo below.
Yeah, and boy, do we have a show for you. So as mentioned, we're doing our top 10 review of 2022, where we look at open access papers that are most cited through 2022. So we do open access because if we can't read it, you can't read it either. And also, it's an opportunity to look at the papers which have had the biggest impact this year that we maybe have not covered so frequently. The two big outbreaks, monkeypox and SARS-CoV-2, we're not going to be covering those so much because those have already been discussed to death. So we want to take a look at the stories that we might not have followed up on, and it's an opportunity to look at kind of what kind of impact these might have had on the field.
Yeah, absolutely. And at the same time, I think the fun thing we were just talking about before the stream, the fun thing about going through the top 10 cited is we get to see, first of all, things that we picked up on in terms of trends in science, and it can feel good. Like for us, I'm like, oh, we saw 3 of these already in previous episodes. But also, it can also show what drives citations in papers, you know, and we can have a bit of discussion maybe on each paper as well, why we think that it got a lot of citations.
Yeah, exactly. And so with that all said and done, we're going to start off with the first paper. This paper got 51 citations, if I recall correctly. Let me check my notes.
51.
Yeah, 51.
Tied for the one number 9 as well.
Yeah, tied with number 9. It takes a current number 10 spot. It's titled Nirsevimab for Prevention of RSV in Healthy Late Preterm and Term Infants.
Yeah. Actually, in the UK, was there it was the same? You guys had an RSV and flu sort of bump after some of the COVID distancing protocols were lifted?
Yeah, that's exactly. Yeah, we have had RSV as one of the things that have been coming up, and I think this paper is, it's very timely in that instance because I think this paper was investigating this treatment for RSV, and then it got interrupted by the pandemic, and then it picked up afterwards.
And so that stands for respiratory syncytial virus, which in my virology classes, it was that's where I learned the definition of a syncytia. And for those of you who have been following our shows, we've seen them before in data. It's when the cells all fuse together in one big blob. And that's because the surface proteins, the fusion proteins, get expressed on the surface of cells. So then infected cells can fuse with other cells. It causes a respiratory illness. And then particularly it's of concern in children. And in this paper, they look at 1,500 or 1,490 infants went through randomization in this one. And they're receiving an antibody that targets specifically that fusion protein. So the idea would be it both stops the virus from fusing into the cells, but also infected cells might also not be able to fuse to other cells with this particular antibody. But they're not looking at the mechanism here. This is a clinical study, so they're looking for outcomes. The primary efficacy endpoint was RSV-associated respiratory infection through 150 days after injection. So they want to see a reduction in how many people got that infection. And then they looked at a secondary endpoint of people actually going to the hospital.
Right. Yeah, and this is very important because I think they did some previous studies with preterm infants, and this is looking at just healthy infants, and the idea of giving them this antibody just while there's an RSV outbreak going on, and
Yep.
so that's why they needed such big numbers because they couldn't guarantee that all the infants would get RSV. So and I think when they did like measure up the numbers, the proportion of infants who got the RSV it was much lower, and it's because they did like a 2-to-1 model, right, where they had like the control group was like 1, and then they provided treatment for twice as many children.
Yep.
Yeah.
Yeah, like because that makes sense because they need they're not infecting children with RSV, right? It's just like if children happen to get RSV, so having that having the experimental group larger is important. Yeah, it seems like out of 12 out of 994 infants got RSV, and 25 of the 496 in the placebo group got it.
Yeah. So so if you break that down to percentages, that turns out to be quite good for, I think it's a 75% efficacy for that. And they did look into the infants that where they still got RSV after the treatment, and what they found was I think 33. That's 12 in the Nirsevimab group. I need to and 21 in the placebo group were infected with RSVA, and none of the patients in the Nirsevimab group got RSVB. So that's an interesting kind of interaction there that could be quite useful for future investigations to tune the drug to be better.
Yeah, I mean, I feel like I never have a lot to say about clinical studies. Like you can see, did we look at what figures have we looked at?
So far, I mean, the fanciest figure is figure 1. That tells the biggest story.
Yeah, and typically the one that folks were like, especially when you're skimming, paying attention to this one, gives you that sense, understanding the figure. Yeah, I think that's all I really want to say about it. I mean, and monoclonal antibodies are definitely like one of the large therapeutic areas that are out there in the world, because we're able to raise antibodies to a variety of different targets, including targets that are just like our own immune things. In this case, the target is the virus itself. So yeah, very popular treatment, and I don't want to say RSV is emerging, right? Like it's been around for a long time, but yeah, for whatever reason, the large spike in it after the case of coming back together, that's I don't know, that's like something that's happened in the world, and I feel like it brought a lot of attention to RSV.
Yeah, exactly. It was very newsworthy. There were lots of news articles about the RSV outbreak that came out, and I think lots of alerts for physicians and people to be aware of it. And I think one of the things that's quite nice for those papers to have is to be able to cite some of the latest research on new treatments. And so often in papers like that, you might see a paper like this being cited in the introduction or discussion, or especially for people who are developing antibody treatments as well, because again, as you mentioned, monoclonal antibodies are a really emerging technology and really important. So if you have
like emerging, they've been emerging for like huge, like quite a long time. It's I would say it's like a very established, but very tailorable. Like that's what I was trying to get at my statement. Like it can be tailored to a variety of different therapeutic reasons, and so yeah, it's that's why it's so popular maybe. And maybe the other part too is that they're usually very it is also a very pricey like a lot of research and development goes into it, so like they are darlings of the pharmaceutical industry world in terms of just like both from the efficacy point of view, which is great, but I mean also from the profits point of view, which is great for them.
Yeah, I mean a preventative antibody is something that I think pharma companies quite like, but it's not necessarily as practical as say a vaccine would be.
Oh yeah, I mean vaccines would be amazing on the preventative side.
I mean, in this sort of case, you are looking at infants where it might the one case where it might not be feasible to do a vaccine. So
Yeah, yeah,
it's
yeah. I
but
mean,
yeah,
this type of work is good. It's necessary. It's necessary to both from the business perspective and from the healthcare perspective. I think it is necessary to explore if you have something that's effective in a certain condition, explore whether it could be effective in a larger population, right? And so you were saying that this was already shown in preterm infants. So now going into the infant population in general, like there are benefits to be had from collecting this data for sure.
yeah, right.
Okay.
Next paper. So this is an So the title of this paper is Investigation into Cases of Hepatitis of Unknown Etiology Among Young Children, Scotland, 1st of January 2022 to 12th of April 2022.
Mm-hmm. Yeah, also 51 citations, so tied with the last one. Also health, but this time we're looking at a report. Actually, this never even hit my news radar, but I guess it hit the news radar of people who are paying attention to infectious disease. It's describing like a small cluster of hepatitis cases that happened in children where they didn't know, they did all the tests, like what virus could it be of all the things that they knew caused hepatitis, and they couldn't, yeah, they didn't find what virus it could be.
Yeah, this is a paper where they discovered like a set of a disease, which is always very important for the field, and they did some work in trying to figure out what virus is causing it, and it seems a bit complicated because in a couple cases they found that an adenovirus may be associated with it. Also, they maybe thought that there was an association with SARS-CoV-2, the two viruses that are already quite popular within the population at this point, let's say. So
All
it's
the rage.
yeah, that so I mean the question is whether like you'd get these sorts of proportions if you just did a random sample of people, and how different that is from this cohort. And I think this has been very popular because it's a new and emerging disease. And once this team announced this case and exactly what the description of it was, other people looked in their own fields of areas of medicine, and they found that other patients had it. So
Yeah,
I
they mentioned like a cluster was also identified in the US, I think, of similar unexplained hepatitis cases amongst children.
Yeah, exactly. I mean, I pulled in like one of the later reviews for it. And I think that they've now found about 1,000 cases of this. 1,000. So the WHO reported 1,010 probable cases of severe acute hepatitis of unknown etiology, including 22 deaths. So one of the things that really stood out was actually how severe this was, because this came up very quickly, and I think some of the children had to like have liver transplants, and it's quite scary because we don't know exactly how it's being transmitted, how it's caught. There are some theories that it's related to the adenovirus, and there have been recommendations of like performing more frequent hand hygiene, avoiding crowded spaces, or basically the standard suite of precautions you get for when you don't know what the disease is.
Yeah, yeah. And
I,
I think at the end of the day, it's reports like these that is informing the medical community, like to keep your eyes peeled. Adding more cases into the investigation helps narrow those down and brings more minds into thinking about like what could be happening here.
yeah, this is a genuine mystery at the moment, which is both. It's going to be something that I think the field will be picking at for a while because it seems like
Yeah.
it's more complicated than this being an unknown virus.
Yeah.
So there are lots of people who are investigating the idea that perhaps it's some kind of SARS-CoV-2, another kind of
Yeah, of
post-SARS-CoV-2
its
disease.
weird sequelae.
Yeah, or there could be an idea that it could be adenovirus associated or associated with adeno-associated viruses. Again, I know that was a terrible sentence to have to say.
But I think the fact of the matter is like there's so much biology going out in the world. It's really hard to pin down like the causative agents of things.
Yeah,
Yeah,
and
especially
fun.
once you run through all the logical right things that you think it could be, then you're left with like what else? Like you can't run all of biology right through the filters. Oh, there are no Star Trek biofilters,
Yeah,
transporter biofilters, right? Like no databases like this that you can go. It's like it's really like your own the scientist wits and like the molecular tools that have been used previously in research. Yeah.
Yeah, another thing I want to highlight is it's going to be much harder to study this because the outbreak peaked in March 2022 and has been in decline since. So it's getting harder to find cases that seem to match this. So there's the possibility that we may never find out what caused this, which
Interesting.
so this is an interesting mystery.
Yeah, I was just looking through like done this top cited episode before in the past, and as I was looking at the one that we did in 2021, and there aren't any like actually we don't often see like these types of clinical reports make it to the list.
Yeah, that's right.
Yeah, like we've covered these reports in the past, especially with SARS-CoV-2. There were tons of reports like this that we looked at. But yeah, we've actually haven't seen one before hit this top citation list. So interesting in that respect, I guess, as well.
Yeah. I think that these sorts of reports, because the papers we pilot here tend to reflect ongoing trends in the field rather than necessarily the content of the reports themselves. So I think this unknown disease is something that is really interesting, especially for. So I think that might have elevated it because I think if they'd found an answer. Also, I think because I tend to filter out things like SARS-CoV-2 and monkeypox.
Yep.
And maybe I should have filtered out this as well, but I legitimately had never heard about this before.
Absolutely. Okay, that's
this
great.
is the sort of thing. Yeah, this is something that I would usually filter out because it would hit all the news, or I'd have heard about it, and I've been kind of, part of this, I'm kind of bored with monkeypox and SARS-CoV-2, so.
I think there is a collective boredom a little bit through it, yeah.
Yeah. So that is this paper. Very interesting unknown disease. Let's see how that goes.
All right. Next paper up with 59 citations. Actually, a lot. This bottom tier is all tied up. So 59 citations. That's the same as number 8. This is Rhizosphere, bacteriome structure and functions. So this is one of these classic overview papers. I think it's a meta-analysis, right? They looked at previously collected data and just trying to figure, so the rhizosphere is specifically the tiny part of the soil ecosystem that's in contact with the plant roots, or also, you could also think of it as bathed in plant exudates, like plants are trying to influence the microbes that live around them. And so, it's commonly thought of that the rhizosphere is a distinct area of the soil. They just want to do like a very comprehensive analysis of bulk soil versus rhizosphere soil and see what sort of organisms live in those categories.
Yeah, I think this is a really important field for agriculture because there's a lot of concern about soils being depleted, and the rhizosphere is like the part that's in contact with plants, the part that really the plants use as a part of their defenses against disease and part of their external digestive system to try and break down minerals and deliver them to the plant. So understanding the health of these soils is becoming more and more important as we try to understand how things like climate change or pollution are going to be affecting that and affecting crops for the future.
But of course, like one of the most complicated things about understanding patterns in microbial communities is there's so many different microbes, and so and everything has like a kind of unique signature because the microbe communities, it's unclear like how common they are across geographies and all these different things. So there's just like a lot of pattern matching, I think, to be done in this sort of field. And so I think the idea behind a paper like this is that it helps us maybe set some sort of pattern to look for, some core set that you might be able to use in your analysis to differentiate. And they do actually, in their study, start making comparisons. Like, so they have rhizospheres from all over the place, so they can group them slightly in different ways. So like, I think they do groupings like, I don't think there's any figure actually that says this. Oh, no, maybe it says cropland, grassland, forest. That's one grouping that was done. But they have some other, I think like the idea of like managed, I think they had some way of looking at like more managed sites versus unmanaged sites. They can look at these sort of patterns, pairwise comparisons.
Yeah, I think they break it down into like different farming types and also looking at the different roles bacteria do in the community, because even in different areas you might find different species, but then you've got processes like denitrification where nitrogen gets released from the soil, or the breakdown of certain compounds. So trying to highlight which bacteria are performing what in which communities.
Yeah.
And whether, or even if sometimes those might not be being done. Like say, like denitrification might be different in certain soils compared to others.
Yeah.
And so I think being able to characterize that and understand, like, oh, maybe this soil for this, being able to like have that baseline ready, so that if you're saying having a soil that is kind of sick, maybe you'd want to go, oh, this is because this bacteria is missing from that community.
Yeah,
And
I think if you take us down to what is this figure? Figure 5 has the one where they added all the sort of what the microbes do functions. And like, I think
yes,
it's important to remember that this is done with 16S data. This whole study was done with 16S amplicon sequencing. So what the data set the data that's collected is identity, sort of this rough idea of identity of microbes. And so how they got all of these functions was there's a database that exists that like matches like identity to functions. And so like that's not the same as previously, like last year, I remember we covered a bunch of papers in the top list looking at MAGs, also maybe throughout the year, right? Metagenome assembled genomes. And so like that's not this, right? They don't actually have view of the genes in this study. This is just like an association through a database of pre-existing associations. Yeah.
yeah. And I think that this is quite useful, I think, to just do that broad characterization. It isn't as detailed as a MAG would be. But
It's not as direct, right? Like
no.
like this is not direct observation of these functions in these communities. It's like inferred. I didn't look into how that database is built, but of course, like that's like a big part of sort of like where the uncertainty comes in. And maybe I'm also like I also want to dive into, because like later on we have like another metagenomics paper that sort of like is looking closely at methods. And so like it is really important. Like I think in this we often like are trying to skim through like a bunch of papers and give people these top level points, but it's also important to remember that those points sometimes are like heavily influenced by the way things were approached. And that's why like we say that if people are more interested, we should do a deep dive, because like doing that deep dive helps us like we'll have more time to really like untangle some of those things.
Yeah, it gives us the opportunity to train our own critical eyes on things. When we do those deep dives, we have to learn a lot of stuff to try and go through papers.
Right.
So yeah.
So yeah, I think it's a really cool paper. Again, because it's the meta-analysis and it's sort of a big zoomed out. I think that. Oh, what's this?
Oh, this is a graph of like citations for Rhizosphere. Because I felt it'd be quite interesting to point out how like much is being published about. Like I think like 1 in 4 papers published about the Rhizosphere was published in the last 2 years. So it is a really like skyrocketing topic. So that was like that. I feel like that explains its place on the list. This is like something that's very popular and very, very current.
Great. Yeah, that I was trying to say what you said very elegantly with showing up some data.
Yeah, it's not as easy for other places but this paper had a very key kind of keyword that I could use to search in a database and find a trend for.
Excellent. Okay. So tied with this one with the same number of citations, 59, we're looking at ginseng polysaccharides alter the gut microbiota and kynurenine/tryptophan ratio, potentiating the antitumor effect of antiprogrammed cell death 1/programmed cell death ligand 1, anti-PD1 and PDL1 immunotherapy.
Thank you for saying that paper title for me. I feel like I appreciate that so much.
No problem.
But yeah, this is a study on ginseng being used in combination with an antibody against a cancer biomarker that has been proposed as something that could be used as an immunotherapy.
That's right. I mean, I think it is. I think it's already in like there are anti-PD1 therapies already approved for cancer therapy. It's sort of one of the more well accepted antibody monoclonal therapies out there. PD1 is like, I think it's like an exhaustion marker, something like this.
Right.
It's expressed by immune cells to say like don't interact with us anymore. To try to like, it dampens immune responses. And I think something about tumor cells, it was found out that like they sometimes express this this ligand to try to just stop immune cells from looking at them. And so it was found like if you blocked it, immune cells may actually start going after cancer. Like the natural capacity of the body to just clean up cancer cells is sort of activated because the cancers can no longer signal to these immune cells that don't look here.
Yeah. And adding into that, they have these ginseng polysaccharides, which are sugars that are being derived from the ginseng plant. So I think this has come from this broad push based in China for trying to turn traditional Chinese medicines into pharmaceuticals.
Yeah.
So the thing that everyone points to is artemisinin, which is a malaria drug that was derived from traditional Chinese medicine, and they're trying to turn their eye on all sorts of different places to try and find out whether they can find the next like drug that they can market and use that they can demonstrate to work. And so these ginseng polysaccharides are an example of that.
Yeah, actually, I think that I just want to say that's a really interesting like research methodology that's being taken on. This idea that there's knowledge to be learned inside of traditional medicine systems, but if we're able to define that within like a more technical sphere, then you can make money off of it in a way that you couldn't when it was just in the traditional medicine area, because then it can enter sort of like the modern pharmaceutical industry. So yeah, one way to approach it, of course, like there's many criticisms for saying like why that might not be the way that we want drugs to be made, but of course it's also a very valuable pipeline to discover things and make them more widely available like around the world.
Yeah, and it's not just China that's engaged, because I remember we've talked about the Ancient Biotics Initiative, where there's some people going through some old like medieval texts, looking at ancient treatments and testing them out to see if they'd work.
Yeah, that was a long time. That was in the original Micro TWJC, right? I remember we covered that.
Yeah, back when we were only on Twitter. So I think that there is some like there's some merit in this, because the idea is that back in the traditional days, they were also iterating through different like treatments, and if there's something that worked, then maybe there might be something to it, and if we should try and investigate further and pull out and see what kind of knowledge we can pull out of that. So usually ethnopharmacology papers, they look at like they're part like sociology and part pharmacology, where they go like, oh, here is how the traditional medicine is used in its original like way, by the original people who use it, and then they look at like kind of examine it and try and test out potential like ways it could work. Because I think there's another thing that people need to be conscious of, is that if you're going to like a community and taking some of their traditional ways and then ending up marketing it, then we need to be sensitive about like actually just pillaging someone else's like culture and then making a profit off of it. So that's kind of something that also needs to be balanced out when you're doing this sort of thing, because but this paper doesn't delve into that at all. This paper is strictly like, oh, we squeezed out some sugars from ginseng. Let's see if we feed them to mice while we're feeding them these monoclonal antibodies. Are there better outcomes?
Yes. And I think they do see a positive effect feeding animals these sugars. And they try to investigate a little bit deeper, like what that effect may be due to. And they link it to like cell type proliferation of some sort, and they link it to microbiome as well.
Yeah, the idea that adding these ginseng polysaccharides changes the microbiome, and that might have a positive effect on the responses to the antibodies, because there is a belief that the microbiota does interface with the immune system, but the question is how that can benefit treatments.
So it doesn't. I feel like, I didn't read deep enough to understand like whether it comes to any conclusion there. So I remember that they did talk about IELs, so like intraepithelial leukocytes, so leukocytes that live in the epithelium. In the gut, there's a bunch of IELs that sit at that interface, and those IELs are constantly looking at the microbiota around there, modulating their behavior on what they see. So I think the hypothesis that these authors are trying to forward is that these ginseng polysaccharides do some sort of modification to the microbiome that then sends signals to the IELs, that then send signals to the rest of the immune system, that makes for whatever happens, because there's so many like whatever happens like domains here, makes for whatever happens when you then get that antibody treatment, the immune system is more active and able to gobble up the cancer that's being sort of made vulnerable by the immunotherapy.
Yeah, they tried to like demonstrate this by they gave some ginseng like kind of so because I think there are people who are being treated with pembrolizumab,
Yep.
and they looked at those in terms of responders and non-responders, and they were like given ginseng, and then they took out the microbiome from those people, and they put them into the mice.
Yep, the notobiotic mice.
And yeah, and then they added, then they did their treatment model to see whether injected tumor cells into them to see how well the mice could protect themselves against.
Yeah, and then for those that were, yeah, basically the responders microbiome like set better results for the mice outcomes.
Yeah, because I have been trying to hold off a little bit of my skepticism of this paper because. But I feel like I have to bring in something that I have with every kind of nutrition-based like treatment paper, which is that the controls never have nutrition. So let's say, because effectively in this paper and other papers, they're feeding mice nutrients. And one of the key things that happen with them when mice get sick is they stop eating. So actually force-feeding mice is a general treatment that can help them survive and help them boost their like their responses to diseases. So I always want to see like I think there are like some papers that have done this where they give just a neutral food source to the mice
as the control,
and then demonstrate efficacy. But I mean, again, so I think a lot of this paper's citations is a reflection of the field itself because. Again,
the excitement around cancer therapies, the excitement around microbiota, and the excitement around taking traditional medicine and turning it into marketable pharmaceuticals.
Yeah, and Chinese medicine is very big at the moment, especially this kind of research into Chinese medicine. I mean, because I think China publishes a lot of research articles, and I think so anything that is quite big and appealing to that area of research can pick up quite a lot of traction. Yeah, and there's a lot because I think China has lots of different Chinese traditional medicines out there because it was very diverse. So there's a lot of different materia medica that they use. So there's a lot of things that people are digging into. So anything that shows any degree of efficacy can be kind of thrown up and said, hey, this is one way that it works. So I think that can be quite interesting for people.
Yeah, and I guess that's all I want to say. I also want to echo that, like, these types of like it's so ambitious in so many ways, like to make a statement about to make a statement about these type of nutrition-based therapeutics, and then also like implicating the mechanism as microbiota. I have like always a lot of skepticism going into reading these things, but in the skim, that's what we saw. Going a deep dive though could reveal that it's not as interesting as it may seem, or the attention given to it is just attention, just attention.
Because at the moment, we're only looking at this research a year out, and it tends to take a while for our own sense of like
consensus to emerge.
Because at the moment, not enough time has really been had for people to take this research and build upon it, and so usually what happens is that there's like a Me Too effect with research where someone finds something amazing, and then another person replicates it, and then that builds up. But sometimes you can get research that people do something amazing, and then nobody can replicate it, and you hear nothing about it forever. And at the moment, we're in the stage where you can't tell the research that can't be replicated to the research that can be replicated. We're just in the cheerleading phase of things.
Totally.
To be honest, I mean, we don't know that this could be replicable. We don't know that. We can't judge it at this point. This is just as valid as anything else we've seen so far.
Okay. So moving on.
Yeah.
Uh, oh, go ahead.
Yeah, this type paper is entitled Microbiome Differential Abundance Methods Produce Different Results Across 38 Datasets. And this has been
Uh-huh.
cited 66 times. And this is juicy.
Yeah, this is the paper that I alluded to when I was trying to dive into the details of the Rhizosphere microbiome. But essentially, the authors of the authors of this paper are trying to expose like some of the there's like there's a lot of complex mathematical models that get used in order to take that 16S data set, right? Same data set as the last time, right? Looking at small little barcode markers that should correlate to microbial identity. There's a lot of different mathematical models that take that huge table of identities across different environments and then tabulate which organisms are differentially abundant in one versus the other, right? Many different ways of skinning that cat, so to speak, through mathematics and computation. And so the authors point out the different debates that have come around these different algorithmic methods, and they set out in their paper to essentially compare them head to head, focusing on this idea of do they find the same abundances between different samples, and also focusing on the idea of if they give samples that they know how many they know what the differential abundance is, like through other methods. What's the false? Let me roll back. The other one is they give like a synthetic data set and look at false discovery rates. So in a data set that's being artificially made so that there shouldn't be any abundances, do some of these algorithms throw up abundances anyways, just in those cases? So yeah, head-to-head on methodology, which makes this paper very difficult to skim and really dive into because it's all in the details essentially of these different algorithms. But I guess there are some broad takeaways that the authors summarize. Do you have something else to add here, Fuzz?
Oh yeah, no, this is like almost like a witch review site of like which of these algorithms work and which of them don't.
Yeah.
And yeah, so they're taking that for a test drive, and there's certain like statistical questions they're focusing on. So when you're out sampling a metagenome, you get different numbers of bacteria just through random chance. And in order to make them comparable, there are certain things they do to the data, like if there's so they go, oh, we'll go for things for a certain sequence depth and discard other samples. Mm-hmm. Yeah, trying to keep things as even and fair between them. And this paper is essentially like a consumer report on like how well they do those, or in which situations things work and which situations they don't. So I mean, I think the fun part is like the last few paragraphs where they start like telling their opinions about certain algorithms, where they say, oh, these ones we don't recommend. They don't work, or these ones are more consistent, and that's kind of the fun part.
Yeah, actually, one of the algorithms they point out, EDGE-R, is actually an algorithm that's typically used for looking at differential abundance of transcripts in RNA-seq. And like you have to imagine, I think a big takeaway that I got from this paper, again, as much as I could without being like a method guru in this area, was that the underlying structure of the data means a lot for the ways that algorithms interact on it. So you can imagine that just like in nature, right? Like there's a structure to the data that underlies microbial samples that may not underlie samples of RNA transcripts. And so they definitely say that using that EDGE-R algorithm, which was developed for a different underlying data set, may not be is not one of the top options to use in these data sets, in microbial ecology data sets. But what I found from reading the discussion was largely it's about understanding what your model is doing in order to make more informed conclusions about your data set. The authors I feel like this paper is really sort of like a little of a red flag warning sign. Slow down if you're becoming a scientist who just wants to slap on the latest algorithm onto their data set and spit out answers. You do have to take some thought to understand what those underlying assumptions are inside of the algorithmic analysis to make sure that what you're concluding is right. Because they do find that using different algorithms on different data sets show different taxa as differentially abundant. And it's not as easy as just looking at the consensus between all of the algorithms either, because that is sort of flattening the statistical realities in a way that isn't helpful. So it is about understanding a little bit of the underlying data, or collaborating with people who do.
Yeah, I can sort of see this coming up in papers where now that this has been published, every single reviewer out there might just be pointing out, oh, by the way, in your model, you need to address this. Or if someone, after this, I imagine that anybody who's been working with EdgeR for the past 4 years during their PhD might be like, oh no.
Got to reanalyze with these with some other algorithms, yeah.
Yeah, and so I think that, and if anyone's doing a new model, they might be able to have to like refer to this methodology to be like, oh, this is what. I think this is the kind of paper that makes the whole rest of the field kind of stand up and go, okay, we need to be a bit more careful. Yeah. I mean, this reminds me of the fMRI papers where they put a salmon in an MRI machine and proved that that with using fMRI you can prove a dead salmon has brainwaves. This has that kind of vibe to it.
Yeah, definitely. I think I mean, I really appreciate that this paper has this attention as well. Like, of course, maybe you can say because they cited all these different methods, of course, right, anyone who's worked with any of these methods, attention is now on it. But it does help to raise consciousness within the scientific community and have that check and balance. Like, part of like scientific rigor, right, is like you try your best yourself, but you always have your own limitations based on your training environment. And then someday you stumble upon this paper, and like that's a big learning moment, and really like just makes you a better scientist going forward if you can digest this. If you are using these tools and you can digest this paper, right? Like this is something that's really helping you as a data analyst.
Yeah, I think I've said everything I need to say about this paper.
Yep, me too.
Right, so the next one is, let's just all pat ourselves on the back here. This paper is titled Tumor-resident intracellular microbiota promotes metastatic colonization in breast cancer, and it's been cited 75 times, and we reported on it previously because
yes, the episode is it's the episode that starts with, if people are searching YouTube, it's treacherous commensal bacteria is what we clickbaited titled this particular paper. Yeah.
I mean, you can just blame me for that title. I'm the one who does them. You don't have to take any responsibility for that.
Oh no, it's fine. I mean, like, we know the medium that we're on, so
yeah,
it is as it is. And yeah, I actually feel like we had pretty good discussion of the paper. Yeah, we had a pretty good discussion of the paper there in that episode. Let's see if I can pull up. I think it was in August. Or sorry, not August. Sorry, April.
Yeah. So yeah, I think it's interesting because I think what I found really interesting is like you've got this symbiosis between cancers and bacteria and these symbiotic bacteria, and then these bacteria can help them like survive shear stress by activating parts of the immune system that cause like that would ordinarily help these cells be more available for the immune system, but actually it makes them survive shear stress so that they can, so on the journey to metastasize, they can survive that journey better.
Yeah. Like I think we discussed it in the last episode too, like just really interesting in terms of like the way in which the bacteria that have co-evolved to live on us then also can find niches inside of cancer cells that help those cancer cells have better survival. So almost like a coevolution at this side as well, right? Like both with the host, but also the host rebels that are growing inside of us.
Yeah, I mean, there are some lovely fish images in there. Like, wait, hold on a minute. How dare they? Do they? It's no longer open access. What?
Oh, really?
Oh my! I didn't know they could do that.
Well, if you go to our Zotero library, we have a PDF that we pulled down from an open access version. So
yeah, link in the doobly-doo to our Zotero. Oh
yeah.
My! How dare they?
Yeah, so they do have a lot of They do have a lot of fish images where they're looking at cancers. They use organoids in this particular paper, so they specifically create organoids out of cancer cells, give them the microbiota that has evolved with them in other systems, then inject those organoids into mice and see how they go, or yeah, see how well those organoids survive under different stress conditions. Yeah, a really interesting paper. Again, we've spoken about it before. And going back to why we think it's heavily cited, well, we chose it. I definitely, when I chose this paper for the docket, I do remember thinking, cancer, people love cancer. It's like such a hot field. Everyone wants to live forever in one way or another, / maybe have known people who have died from this disease, especially in the Western world. And microbiota, which is also an incredibly hot field. We've come to this understanding that we're covered in microbes, and that mediates interactions around us. So it seemed hot for me for those reasons, and I think that that's also why the citations have picked up on it.
Yeah, I looked at the citations. A lot of them were like just the sheer newsworthiness of it. So you see lots of cancer journals suddenly a lot of interest in tumor microbiomes and using that as an avenue for treatment. So I think that this is something that I feel like will be impactful for the field going forward, because this is a key, like, it shows the mechanism for why a microbiome might be very important for tumors.
Yeah.
And that allows people to really cotton onto this and say, yeah, this is not contamination. This is something that we need to be thinking about treatments for.
Right. Okay. So let's move on to our next paper. This one is now looking at 90 citations. Also in the publication Cell, engineered virus-like particles for efficient in vivo delivery of therapeutic proteins.
Yeah, I'm bummed that I missed this one, I think, because this is
yeah,
it's definitely ours sort of thing.
Definitely. It has that application that I feel like sometimes when you're trying to find interesting papers to communicate to people, it helps when you know how it's relevant to human health, because we're all sort of inwardly looking people, and human health is a big part of what people think of as a benefit of science. And so here they're using virus-like particles. So they aren't viruses. They don't have the typical viral payload inside of them. So where a virus would contain the instructions for replicating itself, these instead contain the machinery for doing genetic editing. So for example, the CRISPR ribonuclear protein complex, that is the CRISPR protein that is loaded with a template RNA, you can just have those molecules loaded up in virus particles, and then you have all the benefits of targeting that viral particles have. We've seen, like, through pseudotyping, right? Inside of SARS-CoV-2, you can essentially add whatever receptor you want onto the envelope of certain viruses. And that lets you target it to a variety of areas. And instead of delivering the genetic payload, instead what's delivered is this proteinaceous payload of gene editing tools.
Yeah, that is the most impressive part for me, because usually, we've seen like places where ways to get DNA into cells. So we've seen electroporation where people get stuck with needles that electrocute them, and then that gets DNA into cells. You've got liposomes, got various viruses, but we've never seen something that delivers protein into cells in the same way. And that's really, like, interesting to me, because I think because this actually can edit the genome. This can go into live cells.
I will say, like, I did have an experience during grad school where we were playing around with different CRISPR techniques, and we had looked at these ribonuclear protein complexes and getting them into cells, and we use lipofectamine. So, like, you can just put RNPs into so lipofectamine is a common lipid-based reagent, essentially making fusion micelles and putting proteins inside. So this is like a little step up. First of all, you can make this using cell machinery, right? Viral-like proteins are great because they sort of self-propagate out of cell culture. And then that idea of, like, it's not just lipids on the surface that are helping fuse to cells, it's also these receptors, and you can change that up.
Yeah, I want to talk about that as well, because the way they go about it is very interesting, because they want to, like, in order to make these virus-like particles, they need these viruses to be outside in the like cytoplasm, right? But in order for them to be effective, they have to go into the nucleus of the cell they're infecting. So there's all these clever like iterations where they tie the protease, the sorry, not protease, the
RNP, yeah,
BE, the
the CRISPR
yeah,
RNP.
RNP to the gag protein of the, so it's based off of lentivirus, the outer capsid protein of that, and then that's got a signal that puts it into cytoplasm, and then when it gets cleaved, when the virus infects, it has a target thing that targets to the nucleus. So there's a lot of like really clever iterative like bioengineering things that they do for this paper.
Yeah. And this paper is very much an engineering paper. They try different concentrations of the RNP inside of the viral particles. They try different linkers that can get cleaved in different conditions. They try different sequences of localization, right, to find the ones that get it into the nucleus the best. So yeah, they really are trying to optimize the system and demonstrate and define an optimal configuration that people can start working with in order to pursue, I guess, therapeutic application.
Yeah, they also tested on primary cells from mice and humans. So these are cells that are taken from live, like directly from mice and humans, and tested to see how effective they are. Like off target, like they're looking at how good they are at hitting target, also like how good they are at not destroying other genes that are completely unrelated.
Yeah.
And they found like some very good like results from that.
So
very focused.
Yeah, I feel like we've said a lot of things that seem like, oh, this is a really exciting thing. I think it's important to remember this is also a very incremental thing.
Yeah.
It was known previously that RMPs have these good properties in terms of off-target effects, because the idea is that there's only enough protein around to do some of the changes, and it's not like constantly being produced. So like it's like dosage almost, like they're dosing like a little bit they're finding the optimal dosage range for editing machinery, and then once that machinery has edited, or as it gets I don't know, damaged over time, just like it'll be digested, and then there'll be no way to make more editing machinery, right? Like you're really just delivering the machines needed for if we're imagining it as like our bodies as a construction site, and we're delivering like dump trucks. If we deliver DNA, it would be delivering like factories to make dump trucks continuously to do the work, right? But here we're just delivering the right amount of dump trucks.
Yeah, I think that is a very good way of putting it. You single-use dump truck, whereas
single-use dump truck.
Don't teach a person to make dump trucks if you only need a dump truck once. I mean, we just have to have dump truck. I mean, yeah, we stand waste technicians here, okay? We think that they're an important part of the community and they do an incredible job.
Yeah, I think that's mostly everything I want to say about this paper. I did just want to point out to people this fun technology. I'm not sure if people have heard, but I worked with it a little bit during my job as a technician. In Figure 5, one of the models they use to look at efficacy is P0. So that's neonatal mice still in the uterus. Intracranial injection in the ventricle, in the developing brain of a mouse. Like there's a fluid-filled space that you can inject all sorts of transformation objects into, and and you can get good transformation, and you'll see the results of that transformation as the cortex develops. I want to point that out. I'd worked with a technician who did this technique, and it is crazy finicky. As you can imagine, injecting into the fluid cavity of a developing mouse brain.
Yeah, like I had to do this for like full-grown adult rats, where we have to do injecting a gene therapy into a and you're trying to target a position that is like cool. I mean, I imagine for like an embryo, you're targeting like a needle point kind of area to hit. Whereas, and I was targeting an area that was much bigger than that. I'd say like an area the size of maybe like a rice grain. I was not good at that, so I'm very impressed that you managed to do it with neonatal mice.
Not me, a technician.
Not me. Technician. A technician. Okay, sorry. Yeah. Apologies.
Sorry, not even technician. I think she was like a staff scientist.
Right. Okay. But yeah, we haven't even mentioned the blindness. The fact that you cured blindness in a model of mice, which
Yeah.
again, that's quite a good outcome. So I think this is a good paper for encompassing that whole like kind of through line from the idea to demonstrating in cells to demonstrating in an in vivo model.
Yeah. And yeah, and why is it cited? Because this is sort of like this very hot application area. I will say that like the blindness thing, like that is, there's already a lot of gene therapies being developed in this area, right? First gene therapy approved in humans is for a retinal condition. And so I'm pretty sure that the citations around here is really driven by this idea of gene therapies becoming more widespread. And I do want to highlight that, you know, like having discussions and reading sort of nuanced opinions about this, I think is quite important because because you know, technology is happening with or without us, and like being able to criticize it for why we would want something versus why we wouldn't want something is important. And we haven't done a lot of that, I think, on this channel, but like it is something that I think about on a regular basis.
Yeah, I think so. Because it is getting to the point where people are trying to bring them into medical practice, especially so anytime that someone says, "Oh, we've got an idea for CRISPR that could be used for medical purposes," they can cite this paper and point out that so I feel this is very much like of that introduction bait sort of style of thing.
That's right. That's right. Well, I mean, also, like, as I said, like there's already gene therapies that are being approved in humans, right, for niche conditions. Like we should be having discussions around, right, like who gets to choose which conditions get cured and which doesn't, right? Like what other, like once these drugs are introduced into the environment or into the public, like what about off-label use? What would that mean for people, right? And these are big policy things.
Yeah. Gene doping is another thing that I've heard for Olympics, where like, add a gene to make your athlete like run fast for 10 minutes, like that, like stuff like that. There's because I remember like one of my genetics professors, he was one of my professors was on the committee for trying to detect gene doping in athletes in case like athletes decide to do gene therapy. And at that point in time, that sort of thing wasn't practical. But now it's getting more and more to that stage where we have to be a bit more aware that it is becoming more practical. So, and there are people who might be willing to take the risk of getting cancer in 10 years versus getting a gold medal now.
Yeah, and like, I mean, yeah, I think that like, yeah, just that excitement around talking about it, like it's there and there are people that are doing it. I feel like we don't often touch on it because it's outside the scope of like Microbiology Journal Club. But like, yeah, I just encourage people if they're interested in that type of stuff. Maybe there is one that intersects microbiology. We can talk about it. If you want a deep dive, like let us know. But these are very important conversations to have and to think about. Seek the sources to listen to, not just the headlines, right? Because like in some ways, like the headlines will always say it's an exciting new thing that is poised to save people, but like which people and like also like what other things is it poised to do?
Yeah, I mean this paper, we don't know the side effects of this. This paper is very much like demonstrating the process rather than go. So I mean, there's all sorts of things that practical considerations that we're not at yet. I mean, in terms of like bringing this, I mean, like how much is it going to cost? Who's going to get it?
Yeah, like more societal impact things. I think is sort of just what I wanted to raise in these last few minutes.
Yeah, like red disease, etc. Anyway, should we move on to the next paper?
Absolutely.
Okay.
Taking the number 3 spot with 116 citations is muscle-inspired adhesive antioxidant antibacterial hemostatic composite hydrogel wound dressing via for infected skin wound healing.
So one thing I'd like mention. So this is kind of an interesting. Well, it's an unusual paper for us to cover.
Yeah.
In that,
well, I was surprised that it is up. Well, I was surprised that this was like a top cited paper. I mean, it is. I think it is because. Well, it is microbiology because they're using this like infected skin wound healing model, right? So that's why it comes into the realm of the search that we did. But like in terms of, we've covered this before actually on the show, right? Like there are also microbiology papers that's the significance added to the end, but the bulk of the paper is just talking about creating this material essentially and applying the material in some way.
Yeah, and so the material in this case is a muscle-inspired like hydrogel. So hydrogels are very hot right now. They're kind of they're made of the protein kind of matrices that bond to water, and they're quite important for medical uses, I think.
Yep. And I think in this
And
particular paper, they're using like a polysaccharide, sort of some conjugated polysaccharides to make this particular hydrogel.
yeah, and like, and these polysaccharides conjugated with dopamine, because I think mussels and mollusks use dopamine as like a sticky kind of substance. So we usually think of dopamine as that lovely hormone in your brain that makes you feel good sometimes, but apparently like some organisms use it just to stick to stuff, which is kind of bizarre for my thinking. But yeah, it's and this paper is a lot about how this is being constructed and its properties. So the things that are interesting about it are that as a hydrogel, it means that if you put it over a wound, it can absorb a lot of liquid, and they think that it might also have impacts on the clotting processes, and also might be antibacterial.
I think they try to make it antibacterial by adding zinc into it. So yeah, specifically, like I guess the hydrogel can absorb a lot of different compounds. They're trying to avoid antibiotics, so they instead use metal ions in this particular system.
Yeah, like the silver-infused plasters you can sometimes get in some stores. I think that the rationale for that like makes sense. The only issue I have, well, I've got a couple of issues with this paper. Firstly, it is very, very hard to read. English is clearly not the first. Well, usually, like papers like this would get a once-over by a proofreader to give some help to the authors to translate into English. This probably didn't happen for this paper, and it really is kind of obvious. I mean, there's some things like they mention muscle-inspired adhesive, which you can only find out if you look at the references, because they don't mention it at all during the paper. And some of the models
it didn't feel like a very user-friendly paper from the perspective of like we're not direct peers of these individuals, and so like it's not doing good work in terms of communicating to non-peers, non-direct peers, like what is going on. Which I mean, ostensibly the scientific literature is for other scientists who are studying the same thing, but it also does help a lot, and also from the idea of sort of criticizing the data, like it will help to do good writing and invest in that part of a paper so that, you can have more wide-ranging opinions weighing in on the data that's being collected.
And some of the models they use aren't, well, they're not necessarily very well explained in the group of the paper. I think they're explained a little bit better in the supplementary materials, but there's still things that I'd be looking for, such as like numbers of mice, kind of. And they use
and
like,
numbers,
they use some very like there's some models I'm not familiar with for bleeding. So they have this like thing where they like cut off the tip of the tail of a mouse and then measure how much blood comes out, which I guess for what they're doing it makes sense, but it's not what I would be doing in that sort of situation. That's the sort of thing that I wouldn't be allowed to do without certain precautions from the RX committee, basically.
I guess the models, they don't fit into what you would think of as IUC, or like sort of review board approved, animal review board approved experiments.
Yeah, but I think that's because the British system is very it's very tightly regulated. So there are so we do have to like do a lot more to try and like convince people that this is absolutely necessary. But yeah, aside from they use this model, they put their kind of wound healing salve on that, and they do show that less blood comes out without blood loss compared to say other treatments, or doing nothing. So yeah, but yeah.
It was, I like, like we were saying, we were talking a little bit before the show, like I was surprised that this paper has made it up into the third position in terms of most cited, just because I did not find it easy to read. And so like, it just didn't seem very important to me, like the results that came through. But again, I'm also reading it as sort of like a microbiologist that's interested in mechanism, and maybe this paper has some greater meaning for like the medical biomedicine field, something like that, like some sort of engineering discipline.
I feel like definitely biomaterials. So this is
biomaterials,
bioactive materials. So this might be very important for people who are interested in the biomaterial science of things. So it's not so much about the results, but maybe the techniques they use to crystallize these hydrogels are very unique and important.
Yeah,
They're not,
the photopolymerization and
yes,
yeah, the choice
that
of those
could be,
things.
yeah, that's something that I'm not familiar with at all, but I think that could be very important because I think I've only heard about that in regards to maybe 3D printing or things like that. So that might be something that's very interesting for people in that field for making hydrogels.
But yeah, I don't know. That's all. I'm not super interested in this paper. Just it's not something that I would have gotten excited about if I saw it come through on a list this year. But it was exciting for some population of scientists out there.
Yeah, hydrogels, they're really hot right now. Invest.
Okay. Is that all you want to say about that? Okay, yeah.
That's all I want to say about that.
All right. So another paper that we have looked at before, longitudinal analysis reveals high prevalence of Epstein-Barr virus associated with multiple sclerosis.
Yeah, that's right. We discussed this in January in the paper in the YouTube video titled "Cannabinoids v SARS-CoV-2: Epstein-Barr virus causes MS and the antibiotic apocalypse is already here."
Yeah.
We go for.
Oh, no, I think we did a good discussion on this back then, and it still sort of stands now. Like, it's an impressive paper because it has a large data set to look at a very difficult to pinpoint phenotype because of the prevalence of Epstein-Barr, and yeah, making it difficult to really disambiguate whether or not it associates with MS, which is a very rare disease.
Yeah, like they sampled 10 million people over the course of 20 years based in the military so they could take longitudinal samples. And so they can point out, oh, this person has MS. What do they have EB before or after that? And yeah, they managed, and because they had enough numbers there, they could demonstrate that there was some association there.
Yeah, and I'm really excited for the mechanistic follow-up to this paper. Like, just really like with this sort of epidemiological backing in hand, like what models might we find that explore more deeply the relationship of the immune system and MS, adding in Epstein-Barr and the plethora of effector molecules that these DNA-based, long-term dormant viruses have at their disposal. It's not going to be an easy task to uncover mechanism here, but I'm really excited to see what might come from it.
Yeah, I mean, looking at the citations for it, I found lots of things focusing on MS and B cells, where Epstein-Barr virus integrates in B cells. So looking at the role of B cells in MS infection, but also a lot of post-COVID papers as well, and other diseases that are degenerate. So there is this almost idea that there is a linkage here because I think we mentioned in the show that this paper does link MS to Epstein-Barr virus, but we have to think of that counterfactual that most people who get Epstein-Barr virus don't get MS. So there is something else happening. And I think one of the things that people are focusing on is maybe that thing that's happening is with the immune system. And maybe that in itself is a related disease to what's happening in post-COVID, and other diseases of the immune system are related to viruses.
Yeah, going back to our classic refrain of the immune system's really complicated, and just figuring out discovering very specific signals that you can provide to the immune system and get the same result, but also how that signal gets modulated by other environmental conditions. And in this case, we could call an environmental condition Epstein-Barr infections, given that there's a significant amount of people holding it right dormant within their bodies.
Yeah, and also there are people who've been working on Epstein-Barr virus vaccines who've suddenly got a massive boost because this was published. Because suddenly Epstein-Barr virus vaccines, they can list that, oh, here's another condition that can be prevented by Epstein-Barr by doing this vaccine. So I think this put a lot of wind in the sails of those investigators. And a whole set of new potential biomarkers to look out for people studying MS.
So again, more discussion if you check out that other episode. So moving on to the most cited paper of 2022, non-COVID, non-monkeypox, open access. We have with 1,046 citations, global burden of bacterial antimicrobial resistance in 2019, a systematic analysis. Faz, when you read the title of the last paper like that we covered previously, this paper was also in that episode as the
Yeah,
antimicrobial apocalypse.
yeah, the antibiotic apocalypse is already here. Yeah, we got the top two papers in the same show in January, so I'm very impressed with that show.
I like that. To me, it was like a personal ego boost. I felt like I was like, my fingers are on the pulse of the scientific community.
Yeah, like what we do, we are getting better at picking out interesting stories, and I'm hoping that we can still continue to do that over the next year.
Yeah, but of
So this paper,
course, I
yeah,
feel like people should throw us. You know, we're only made better by other people's suggestions. So if people are seeing things on their radar and they think they're really interesting, and more people should be listening to or hear coverage about these papers, please toot them out to us, because I think that that's like, yeah, that's why we exist. That's why we're holding space like this.
yeah, this paper like pulling reports from all over the world
Hmm.
to kind of categorize like all the infections that have been happening that were due to antimicrobial resistance, and then doing that idea of like what would have happened to these infections if they weren't antimicrobial resistant,
Yeah.
and then based on that could give us an actual concrete number about how many people are dying from antibiotic resistant bacteria.
And like that's such an important thing. Faz, you mentioned that just before the show. Like, it's such an important thing to have these numbers. It informs policy. It gives people this concrete idea of like the burden, right? Like who's getting affected by this. And you can tell in the discussion of this paper, their eye is on policy. So while this paper probably got tons of citations from all the microbiologists working on antimicrobial resistance saying like, this is why it's an important problem, the authors, I think, like are looking for this information to inform policy and really thinking about like, where are we taking steps systematically in the societies of the world to prevent antimicrobial resistance from spreading? And I did try to read the discussion a little bit more in more detail this time, and I was impressed by one thing that they pulled out. They said just limiting the use of antimicrobials, antibiotics, isn't necessarily what the paper is suggesting, because they're saying that in places, in low resource areas, they're seeing a lot of burden of this infection, but possibly it's because that those regions only have access to the most like second line antibiotics, right? Meaning that subtherapeutic levels or not the best antibiotic is being used in certain cases, right? That could be a driver of why antibiotic resistance comes from certain areas and not others, right? Low resource areas and not high resource areas.
Yeah, and I think it's another illustration of how we need to be thinking globally when fighting viruses, because
Hmm.
viruses, bacteria, microbes, they don't respect borders,
Hmm.
and yeah, the fact that these health systems are being neglected, that we could improve sanitation, because I think I mentioned during that show, I think I hopped on quite a lot about how that antibiotics were used as a stopgap solution
Yes.
when sanitation isn't available. So
Hmm.
trying to improve the whole rest of the medical system is so important. Um,
Yeah.
uh, yeah,
But that's everything I have to say. This is like a, oh, I don't know. I do want to say one more thing about this paper. Um, I actually met like a scientist who worked on it, and they're mentioned in the acknowledgments. I just want to point out that this is a huge collaboration. Like if you look
hmm,
at the antimicrobial resistance collaborators in this section, like there are so many scientists named, and like it's a whole, like it's a huge list of people that worked on this. The data set and sort of the scope of data that's required to bring this sort of paper together is huge. And the scientists told me that they will be publishing like updates, right, to this analysis over and over again. So one really exciting thing about this paper is it really is almost just the beginning of like a really robust network of scientists working together to update the values of this going forward, right? And surveillance in general for antimicrobial resistance. More than just the paper itself, it's a very, I'm happy to have this as our top side paper and bring more attention to it, because it shows like that international collaboration is important, that we need like these systems in play in order to better argue for the importance of different policy interventions when thinking about global health. So yeah.
yeah, no, I fully agree with that. I think having one unified voice to speak on behalf of all the people suffering from antibiotic resistance is so important, because it can get lost in the noise. It's been going under the radar because it's not just one disease. It's lots of little diseases.
Yeah.
And the thing is, the same goes with cancer. There are lots of different cancers out there, and they unified under one thing and said, "Hey, we just need to fight all cancers at once."
But they have a
huge
huge voice that like that impacts the citations in microbiology. You know what I mean? So like, like I'm always happy to see like stuff that's like deeply in the microbiology discipline, like being able to create a lot of noise and argue for what's important that scientists think is important in terms of human health.
Yeah, and this paper they estimated between like I think 1.5 million to 4 million people are dying of antibiotic resistance, which is basically in the realm of like between half a SARS-CoV-2 outbreak to like a SARS-CoV-2 outbreak that we're not noticing, which is it is very important and people need to pay attention to it. And I'm speaking mostly to governments because this isn't really something that individuals can do much about. It's something that has to be done on the health system level.
Yeah, we're talking to you, government officials that have tuned into this YouTube channel.
Thank you, government officials, for tuning in.
All right, well, join us in 2 weeks where we'll cover more cool microbiology news.
Yeah, that's right. And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or comments, please let us know down below or through Mastodon or through Twitter.
Yep, I totally agree. As Faz said, Mastodon, Twitter are great places to reach out to us. We both believe that peer review is a process that requires folks to have read the papers. And so if you think you have something to add or found something unclear, just let us know in the comments or through any of those channels.
It's been a pleasure chatting with you, Danny.
Same here, Faz.
Tune in 2 weeks for more microbiology content. See you soon.
Bye-bye.